Okay, the purpose of this training is to walk you through the steps for identification of Streptococcus species, and this is Strep Unknown 1. I'm calling this student training module. So here are your objectives. You're going to watch a video on your own showing the collection of the specimen. Streptococcus is typically in the throat. You've heard of strep throat. So that's a good place to swab, to collect it. And then you will learn the biochemical tests and the other physical characteristics that we use to identify what species of Streptococcus you have isolated. So you will, on your own, at some point, go back into this PowerPoint and watch the throat swab video. But once the throat swab has been done, you're going to streak it onto a blood auger plate, just like we did with the staph specimen. The blood auger will show us what kind of hemolysis this particular strep species has. So remember, there's alpha hemolysis, beta hemolysis, and gamma hemolysis, which means no hemolysis. The samples have to be grown in a candle jar, and so you put the samples in this jar, and so you put the plates in the jar and you light the candle and you screw the cap on, and that candle will remove most but not all of the oxygen, so it puts it in a very low oxygen environment, and strep grows well in a low oxygen environment. It doesn't have to be zero oxygen, but low oxygen. So let's say you did the streak and then you grew it on the plate, and you might get a mixed sample on the plate, and what we're looking for is a sample that can grow on the Columbia nalidixic acid agar. So remember, this nalidixic acid is an antibiotic that prevents any enteric gram-negative bacteria from being able to grow. So we want to select for gram-positives, and you might get a result like this. On this plate shown on the lower right of the screen, you see some colonies. Some of them are showing the alpha hemolysis. So these ones that look kind of greenish brownish, that's example of alpha hemolysis. The ones that have this bright clear space around them, almost like a glowing, but really it's a clearing, a lysis of the red blood cells in the agar, that is the beta hemolysis. And in this student training video, we're going to look at the beta, a sample that's beta hemolytic. So let's say you have your beta hemolytic colony, you have transferred it to a new blood auger plate by itself. You grow that up in the candle jar, and then you do a gram stain. And with the gram stain, Streptococcus is going to be a gram-positive, so you see the gram-positive, the purple here. You will see the cocci shape, and you will see the chains. Now, just because you see some singles, those are ones that have fallen off of the chains, but when you see the chain structures at all, that means that you have the strep, and sometimes I think of the word strip like a chain, like a little strip of cells, but really it's lots of cells linked together in this chain, like a beaded necklace. So you definitely have the Streptococcus typical arrangement. You have the cocci and you have gram-positive. So that would confirm that you're probably on the right track for finding Streptococcus. So in this student training, we're going to pick a beta hemolytic colony off of that very first blood agar plate. And what we're going to do are some antibiotic tests. And there's really 3 antibiotic tests you can use with strep: bacitracin. You may remember bacitracin. And then some that we haven't talked about before, but SXT, sulfamethoazole, and then also something called Optochin. We're not really going to worry about why we would use one over the other, but you need to be able to remember how to do the antibiotic test. That's where you spread the sample all over the plate, covering every part of it, and then you drop those antibiotic discs on, and then you incubate. Of course, with the strep, we always have to incubate inside the candle jar. So we would put the plates inside the candle jar, and then I would put the whole candle jar into the incubator at 37 degrees. So let's say that you did that, and this little disc A represents the bacitracin, and this SXT is the SXT. So let's say these were your results. So on the bacitracin, you do see a good zone of inhibition, 20 millimeters, and that's going to be enough to mean that your sample is sensitive to the bacitracin. But your sample is resistant to the SXT because it grew right up to that disc. So sensitive to bacitracin, resistant to SXT. And you want to be writing these down, like when you do the student practice PowerPoints, you can write down what the results are. So on this one, the result would be sensitive to bacitracin, resistant to SXT. Now the catalase test. You may remember the catalase test when we talked about staph. You put a little smear of the colonies onto a glass slide, and then you drop some liquid hydrogen peroxide on it. And in this picture, there's no bubbling. It's hard to tell, unless you're seeing it live in person, but this picture represents no bubbling. So you just see a plain drop of hydrogen peroxide on a little smear of bacteria, and it's not bubbling. So that would be a catalase negative. The next test is called the bile esculin slant. It's an agar slant, and it's kind of this color here. Usually it's a little more light golden, but we'll go with the picture that she put here. And the slant is selective because it has bile in the agar. And bile is kind of a nasty secretion of the human body. And only very few organisms can grow with bile present. And so if the organism can grow, then it's positive for the bile test portion of this. But there's a second test also. If it does grow on the surface, you remember how to inoculate the surface of a slant. So if it does grow on the slant surface, then if it turns the agar like a really dark, what we usually call coffee or chocolate brown, then it's esculin positive. So in this picture, it really looks the same before and after. And so it's definitely an esculin negative on this picture. And she doesn't tell you. I would tell you whether or not there's growth, because the way the picture is here, I really can't tell if it actually grew on the bile. So bile positive or negative, that one's hard to tell on this picture. I would probably just tell you that result. But the esculin, you would be looking for a much darker color in the after incubation picture. So this one's esculin negative. And then we have just some salt broth. And if you inoculate some salt broth, sodium chloride, if it can grow, it'll get cloudy after incubation. If it doesn't grow, it stays clear. In this example, she's saying no growth in the salt. So you've written down all these results, and then you get to this chart. It's similar for the staph. So which test results do we have? Well, in this unknown number 1, we picked a beta hemolytic. So the first 4 here, the first 4 boxes, include a possibility of beta hemolytic. So knowing that our specimen is beta hemolytic, we can eliminate these last couple boxes. So right away we can eliminate those because we only picked, remember, the beta hemolytic had that glowing or clear spot around the colonies on the blood plate. All right. Now what else? We saw that it was bacitracin sensitive, remember, on that plate, but SXT resistant. And we didn't do the optochin on this in this unknown. Bile, escalin, and sodium chloride all negative. That matches. The other species that are possibly beta hemolytic would all be resistant to the bacitracin. So really based on that, and some of these other results, honestly, the combination of results that matches what's presented in this PowerPoint would be this. So Streptococcus pyogenes would be your best conclusion because it matches. So that's what I'd be asking you to do with the next 2 PowerPoints. There's an unknown number 2 and an unknown number 3. And once you've gone through those, if you have any questions, shoot those out to me. And then within the module, you can see if you've identified it correctly. So try to do the identification on your own and then check with the little quiz, with the little module, check and see if you got the right answer. Okay. The main point is to figure out how to do this process so that you can do it on the practical. The chart would be provided on the practical. You don't need to memorize this chart. But you do need to know how to read the tests, what the tests are really about, and then how to use this chart to narrow down to your species.